This is a brief introduction of PrismEXP. PrismEXP is an algorithm that tries to annotate genes with gene annotations. So these can be biological processes or pathways or potentially upstream kinases or transcription factors. The overview of the algorithm is that we try to leverage gene-gene co-expression data, and specifically PrismEXP relies on the ArchES4 gene expression database. So this database contains about 1 million publicly available gene expression profiles for mouse and human. And so it really represents a huge wealth of information of how genes interact with each other. The issue is if we would take these large data sets and then calculate a global gene-gene co-expression matrix, then we really simplify the view of the complexity of the gene expression space. So PrismEXP tries to take advantage of the Simpson's paradox to build a rich feature space for a machine learning algorithm to do these gene predictions, these gene annotation predictions. The predictions that are made by PrismEXP outperform the predictions made by global correlation matrices that are calculated across all tissues at once. So the workflow is shown in this graph here. So there's basically 5 main steps. The first step is clustering the gene expression data. Clustering means we want to identify samples that look similar, and that means they probably come from the same tissue or the same cell line, so they have something in common. And then we calculate the correlation of pairs of genes within each of these clusters. And practically, PrismEXP can take 100 clusters or maybe 300 clusters or 1,000 clusters and then build the same amount of correlation matrices. So these correlation matrices then get condensed into a feature space that is then used in the light GBM machine learning algorithm, and it tries to predict a gene and its annotation. And for that, it's using known annotations. In general, what I can mention here is that overfitting the model is fairly hard because it is only like a semi guided algorithm, so it doesn't really know about the genes themselves, and it doesn't have the full gene expression profile to work with. It only has these features. So this is a good feature in this model. And the good thing is you can train PrismEXP on one gene set library and then apply the model that you trained on other gene set libraries. So that usually works fairly well. The Simpson's paradox is kind of depicted in this plot to make it quite simple. So here we have 2 variables, x and y. And in the context of PrismEXP, these would be genes. So we have 2 genes, and then each point would represent a gene expression sample. And if we look at the global relationship between these 2 variables, they're negative. They have a negative correlation. But we can see in this made-up example that if we look at these 5 clusters individually, they all have a positive correlation. So it's kind of unintuitive, but subpopulations can have a completely different trend than the global correlation. And this is an important feature that we want to exploit. So you can imagine that in one tissue type, 2 genes might be negatively correlated, while in another tissue type, they're positively correlated. And this is generally a much richer feature space to understand the function of a gene and to annotate it with interesting biological concepts. Yeah, so as I mentioned before, you can run PrismEXP on your own. So there's a Python package that you can use, and it should run on a fairly well-equipped desktop computer. You most likely will need more than 20 gigabytes of memory, so this is one of the limitations. And it takes quite a while to finish a prediction run. The hardest thing to do is build all the correlation matrices. After that, all the other steps, they can be run fairly efficiently. So then doing a lot of predictions is fairly easy after that. As a take-home message, what we can show here, what's shown in the plot, that the prediction quality increases with the number of clusters that are used. So the clusters are equivalent to the number of correlation matrices that are used in this approach. And as we can see that if we use, let's say, 300 clusters, this is as far as we took it, that produces the best results, but the improvements do start to become marginal. So increasing it to something really large won't really benefit you at a certain point. But potentially with ArcGIS-4 growing in data, potentially this might also improve the algorithm, and the number of clusters might benefit from having a higher number as well. So PrismEXP, how can you make your own predictions? There's 3 ways to interact with PrismEXP. One is directed for people that are comfortable using programming. There is a Python library in GitHub, and you can just install it on your computer, and it should work pretty well. The other ones are basically interactive user interfaces, and everybody should be able to use those fairly easily. One is the PrismEXP website, which has pre-computed information of genes, and then the other one are appeters that allow interactive input from a user, and then it produces a prediction for a gene of interest and a gene set library of interest. Okay, so if we look at the code, there is really 4 steps. So here's a Jupyter notebook. So the first step is we calculate the correlation matrices, then we calculate the features, then we train the model, and then we make predictions. So that's quite easy. This is the code you would run if you want to install directly from GitHub. And this is also all explained in the GitHub repository. So there's a lot of examples how you can run it. And so the things you would have to really consider is that you have enough disk space. Each correlation matrix can be 3 gigabytes in size. So keep that in mind. So if you have 100 clusters, you need 300 gigabytes of disk space. Since PrismEXP is fairly memory hungry, ideally you have 64 gigabytes. That's the machine that I used, and I never run into issues with memory. If you have less memory, there might be problems. The compute time is significant, especially depending on the number of clusters. But PrismEXP relies heavily on multi-threading, so it's really trying to be optimized in speed, and usually you can run this in a couple of hours. So if you run it overnight, it should be able to finish a prediction and build all the correlation matrices at once. And this is all the code you need. So here what we do is we import 2 libraries. We import the PrismEXP library when we installed it with pip prior, and we need this library to download the ArcGIS-4 gene expression data. So here we download the human gene expression data. And so in PrismEXP, you basically have to set a work directory. And so this would be specific for the project that you want to use. And in the folder, it will store all the correlation matrices, the model that you train, the feature matrices that you build, and the prediction matrices that you build. And then you also want to specify a GMT file, which is a gene set library from Enricher. So the PrismEXP package allows you to download all of the Enricher gene set libraries, and you can basically look them up either in Enricher, or there's also a function that allows you to list all the available libraries in this package. Then you specify the cluster number. In this case, I want to split or stratify the gene expression data into 100 clusters. And then I do my 4 steps. I create the correlation matrices. Then I calculate the feature space. So this is kind of condensing the information using the GMT file and the correlation matrices. Then I train the model. And then I predict based on the existing features. And the trained model that we had before. And because I don't specify them here, it will look in the work directory and then use the feature matrices and the model that are in that folder. So the thing is, in this example, we train on GoBP, the biological processes. We train the model on this because we build the features using this. And then we predict the GMT again, right? So it's a little bit circular. We train the data on the same GMT file. You don't have to do that. So you could train the model on GoBP and then apply it on other gene set libraries. So this is what we did in the paper. And we can show that this works well. But you would get better results if you train the model on the library that you predict. In the case that you just want to reuse a trained model, you obviously don't have to recompute the correlation matrices. What we have to do is, if we want to choose a new gene set library to predict, we have to build new feature matrices and then run the prediction algorithm. So this is kind of shown here. So let's say now we switch, after we trained the model on GoBP, now we switch to KEGG 2021 human, and we now calculate the features for this gene set library, and it will override existing feature matrices, and then it will run the prediction. And so this step is pretty quick, so you can expect this to run in a couple of minutes. So much faster than calculating the whole correlation matrix and clustering and this sort of stuff. So this is how you interact with PrismEXP programmatically, and there's more details on variables and parameters that you can use in these functions on the GitHub repository information. So you can read that in more detail if you want. This is the PrismEXP website with pre-computed annotations for genes. So it's a quite easy to use tool. So here you have as input a gene symbol, and you have the submit button to get the results. So in this example, we can just use one of the two genes that we can just click on. So now the loading process is pretty quick because we just have to fetch existing results. Here you have a list of pre-computed gene set libraries that we're using. And you can see these are table formats where we have the ranked annotations for the gene. So specific things to note is that we have the gene AUC, which tells us how well we could fetch known annotations for this gene in this given library. And this is the AUC value, and you get more information if you hover over this question mark. And you can see in turquoise the annotations that were previously known. So we can see here in this example that in the top 10 predictions, there's a bunch of upstream transcription factors that were already known to be regulating SOX2. And we can go down and look at other tables. So this is the gene ontology biological process library. You can see that the top 2 predictions are already known, even though they're quite general, like regulation of gene expression. It's a quite general term. And then if you just want to download the whole thing, you can just click on this button and we'll download the whole table into a text file, so you can analyze it on your computer. And if you're curious, you can also just click on this Jupyter notebook. And as we've shown before, this is kind of showing the workflow that was used to compute these predictions. In this case, it's just showing the example of the gene ontology biological process. And we've talked about this before, so there's some extra parameters. Then the other way you can interact with PrismEXP is by using epiters. So here is also a link to that tool, and it allows you to get predictions for every gene set library in Enricher. So there's nearly 200 gene set libraries with different types of annotations for genes. It's a very comprehensive resource. And because these are not pre-computed, you can fetch them for a gene of interest. So if we just scroll down and let's say we want to find Let me see if I find something that is interesting. Let's say we want to do the KEGG human pathways, and now I submit. And we can see that we now search SOX2 for the KEGG biological process and the pathways, and now we just have to wait. So the process is a bit slower because we have to fetch all the correlation data from a database, and then we can run a prediction, which goes pretty quickly. So now I can speed that up, and then once the results are in, we can talk about what we get. All right, so now after 2 minutes and having some coffee, we can keep going. So now we have all the gene correlations loaded into a matrix, and from this we can just retrieve the average correlation score. So these are the features that we're using for the machine learning algorithm. And then these steps, they just go instantly, right? So now we can load the PrismEXP model. This is the same one that was used on the PrismEXP website for the pre-computed results. And so now we can look at the prediction validation score, and we can see that from the known KEGG pathways, we could retrieve them pretty well, right? So the AUC is 0.98. But there were not too many known annotations for SOX2 in that pathway database. So the interesting ones will potentially be the ones that were not previously known. So here we go. We have a prediction matrix. And so just to mention how these values come together. So the predictions, this is the actual prediction score. They get z-score normalized based on the population of all the predictions. So here we have a z-score, if you want, of this first column. And we can try to calculate a p-value from that, but really what we use is a normal distribution and then give it these z-scores, which ends in these p-values. And then if we correct for them, they're not always significant. I wouldn't worry about the significance too much. The predictions are more meant to be a ranking of potential annotations rather than really having a statistical proof that they are really true annotations for the gene. And finally, we have 3 different files that you can download. You can just download this table that we see here completely as a text file. And then you can also get the PDF of the plot of this plot here. And you can get that as PDF or as a PNG. And yeah, this is it. So the Aperture, in conclusion, it's pretty powerful because you can run it on every gene, or you can run it on every gene set library. It just takes a little bit of time to get the results. And the pre-computed ones, if you just want to have a quick look, will give you a quick overview of the gene and some of popular gene set libraries that we have. I hope PrismEXP is useful for you, and that's all I had to show for you. So thanks, and good luck with your research.